Hey guys, so this is my last video for right now, and it's going to be on the immune system. And this is, I think, one of the coolest systems, also in a way sort of the most complicated. You don't need to know too much of the specifics, but again, general idea, big ideas in this exam. So, so in the immune system, the idea is you have a pathogen, something bad entering the cell, and your body wants to know what to do with it. So we have a bunch of different cells that do a bunch of different things with this. Um, so you have the exposure to the antigen, right? The antigen, the thing that you're trying to stop. Um, and so you have the antigens will be captured by certain cells, right? And those cells will activate what we call the helper T cells. And these helper T cells are very helpful, right? And this is a positive feedback loop. Okay, so I want you to see what this thing is, right? The immune system, that's somewhere that we would want. We would definitely want positive feedback, right? Because we want that disease gone very, very, very fast, right? I'm sure many of you are feeling that right now. Um, and so we want this disease gone, right? So the helper T cell, it's going to activate both of the big immune systems that we want activated, right? It will help activate these B cells, the humoral immune system, or the humoral immune system, I believe, and the cytotoxic T cells, um, often called the killer T cells, um, that will be part of the cell-mediated immune response. We'll talk about those in a minute. But notice the helper T cells also activate helper T cells, right? So what they're going to do is they're just going to send out a bunch of different signals that are going to activate all of these cells and activate more helper T cells, right? So a helper T cell gets activated and will activate more helper T cells, which will each activate more helper T cells, which will each activate more helper T cells. And so it's positive feedback, and then all of those helper T cells will activate these B cells and the cytotoxic T cells. And so the B cells then can split into one of two things: plasma cells, right, which are going to secrete antibodies. They're going to release the antibodies. Antibodies are what kills antigens, right? Antibodies get rid of antigens. And our whole thing is getting rid of the antigen, getting rid of that disease, right? The memory B cells are also very important, right? Because those uh, hold in those antibodies. They make the antibodies the things that can kill the cells, right? But um, every disease that comes in has its own antigens, right? And so you want the body to remember once it's been infected, and your helper T cell, sort of what it does is it makes a mold of the antigen that it gets from the disease. It makes a mold of those antigens and releases them out. That tells the cytotoxic T cells what to kill, and the B cells what to also kill, in a slightly different way. So the plasma cells don't like directly battle the disease, but it sends out signals. It sends out the antibodies that can help defeat the disease. The memory B cells take those antibodies that it was given, right, by the B cells through this chain of events, and what it does is it keeps them, right? So if that disease ever comes back, this process doesn't all have to happen. This is hopefully not slow, but it can happen a lot faster because we already have on file, right? We already have the antigen on file, so we can battle this disease, right? Once we're affected, we have it in our memory. We know what the antigen looks like, so we can battle it really, really fast, and it can start this process a lot faster. Right? Same thing with those memory T cells, right? Same idea of remembering the disease so that we can keep that we can help fight it, right, without having to go through this whole process. The T cells also have those antibodies, but instead of releasing the antibodies that kill and defend against the pathogen, it immediately it has the active killer T cells. Those just go right in and fight the disease, right? These are your battling cells. And it's all possible because you have to have, there's a specific antibody that you need to fight the disease, gets caught by the helper T cell. It makes that antibody, sends it out to all of these cells that each do their job. And then you remember it, you send out those antibodies into the cytoplasm, into the rest of the cell, or not into the cytoplasm, into the extracellular fluid and the stuff inside, or inside the organism, but not just inside each cell, right? That's how it's going to travel through the organism and get to other cells. So and get to other parts of the organism and kill the pathogen. Hopefully that makes sense, right? It's releasing it out into the body of the organism, so that it can fight the disease, right, without having like cell to cell combat in a way. It's not exactly like combat, but hopefully you get the point. And so then the T cells are the ones that really fight that battle, you know, cell to cell, right? This is just sort of releasing the antibodies, releasing sort of that killing mechanism for the antigen. Okay, so now let's look at the 2018 AP free response question 2. We talked about this one before, but we're going to go in a little bit more depth. I'm not going to answer the entire question, just the ones that really pertain to a lot of what we've been doing, and a lot of what could be on the AP. And so let's we'll read this, and then I'll read the specific questions. I didn't put it all on here because I'm sort of picking and choosing what we answer, but you can click this link and I'll have the document in the description. Okay, so some pathogenic bacteria enter cells, replicate and spread to other cells, causing illness in the host organism. Host cells respond to those infections in a number of ways, one of which involves activating particular enzymatic pathways. Right, here's sort of our signaling pathway. Cells normally produce a steady supply of inactive caspase-1 protein. In response to intracellular pathogens, the inactive caspase-1 is cleaved and forms an inactive caspase-1. In active caspase-1. Active caspase-1 can cleave into two other proteins. When caspase-1 cleaves an inactive interleukin, the active portion of the interleukin is released from the cell, right? So, and then it also affects the gasdermin, and activates it so that it makes these pores, right? I know it's a lot of words, but the big idea is understanding what it does, and it gives us a diagram. Thankfully, we don't just have to kind of figure out what all these words mean. And so what you're having is you have an infection. It activates this protein. This protein cleaves and activates this protein and activates this protein, right? And this one is going to release interleukin, which is going to signal for the immune response to happen, right? And it's also going to open up a pore in the cell. And I wonder what those pores do, and this question will ask us a little bit about that. So the first thing is, describe the effect of inhibiting step 3 on the formation of pores and the release of interleukin from the cell. Right? Well, if you inhibit step 3, what's going to happen? Right? Well, first thing is, what happens to the interleukin? Nothing. Right? Nothing is going to happen to the interleukin. If you mess with step 3, it won't affect step 2. Leukin. No, it's a little bad handwriting, but I think it's okay. Okay, so what happens to the pores? Well, if you inhibit step 3, the pores will not be pores. Wow, I can't believe I made that mistake. Let's see, por. Pores will not form. right? There's no change to the interleukin. It still gets released, right? So this is still released. Okay? And then pores will not form. Okay. Okay, let's look at the next part. Make a claim about how cleaving inactive caspase 1 results in activation of caspase 1. A student claims that pre-infection production of inactive precursors shortens the response time of a cell to bacterial infection. Provide one reason to support the student's claim. Okay, so that's a little bit difficult to just hear. So let's break that down. Okay, so if we what we're doing is we're breaking this inactive caspase 1 apart to make a active caspase 1, right? And so what is that going to do? Well, think about this is a protein. Think about what might, if you get rid of something, how that could activate a protein. Well, we know that there are things called inhibitors, right? Inhibitors are things that when you take it away from the protein, that the protein starts working, right? So we're going to answer that question with this. This is probably going to be inhibitors, an inhibitor, right? If it has it when it's inactive and gets rid of it to be active, that's going to be an inhibitor. So that's something that that question is going to ask us. And what that will do is that will change the shape of the protein, right? Okay. We're skipping part C. Okay, part D. Describe the most likely effect of Gerstermann pore formation on water balance in the cell in a hypotonic environment. Hypotonic means you don't have a lot of solutes outside of the cell, right? It's a hypotonic environment. Well, if this is sort of like, this is close to just pure water, right? Close to pure water. That's what hypotonic will mean, right? Well, what will happen when these pores open? Well, think about water potential, right? The pure water is going to flow into the cell, right? If it's completely uncontrolled, right? So when it opens these pores, you're going to have water flow into the cell. The cell is going to go through lysis and pop, right? The cell is going to pop. Which is right going through lysis. You'd probably want to write that on the AP exam, right? So but that's not exactly, sorry, I got ahead of myself. Part D is just what will happen, and the answer is the water is going to enter the cell. Part E is explain how the pore formation and interleukin release contribute to an organism's defense against a bacterial pathogen. Well, if water enters the cell and it goes through lysis, right? Well, what can the cell not do anymore? Can't replicate. And if it's infected, you don't want the cell replicating. You want it to die, right? In the interleukin, when it releases that gets other cells involved in this process of starting to help fight this disease, right? So this is a way of signaling other cells to fight the disease, right? Interleukin, it says, is a signaling molecule that can activate the immune response, right? So that's what the point of releasing the interleukin is. And then you want to kill the infected cell so it doesn't start infecting other cells, right? So that is the immune system. That is the big ideas of the immune system, and we've worked out one of these AP Bio questions.